pull up a chair, take out the oxygen, and join the discussion.
You've just stepped into Life Unculturable,
the world where conversations grow faster than colonies.
We're your hosts, Sam and Julia, scientists by day,
Podcasters by night,
and professional oxygen avoiders 24/7. Join us in exploring the science of microbes that thrive in the absence of oxygen on Life Unculturable. Wow. Well, Julia, it's great to be here. How are you feeling today?
I'm very excited to be here with you today.
Yeah, well, I'm excited to be here too. This is pretty different for us. We're not in the lab. We've traded in our lab coats for keys to the podcast studio.
So we're sitting in another chamber.
That's true. Yeah, we're in the podcast chamber currently. Thankfully, no airlock, no vacuum seals.
And none of that sound.
Yes, no compressed gas tanks. Yes.
So what are we doing today, Sam?
Well, today we're going to be we're going to be recording the first episode of our new podcast, Life Unculturable. And so in this podcast, I think what we're going to try to do, hopefully, is to chat about all things microbiology, but hone in on some of our specific biases and preferences and talk about anaerobes.
We totally are, given that anaerobic microbiology might be a very underrated topic in the podcast world.
That's right. This probably makes us number one on the charts as it is. We're going to debut at number one, baby. So, Julia, tell me a little bit about yourself. Where are you from, and how did you wind up in Oklahoma?
So I'm originally from Austria, which is a small country south of Germany, for those of you who might not know. And that's also where I did all my academic career before coming here to Oklahoma. I did my PhD at the University of Innsbruck, which is a little town in the Alps. And then I came to Oklahoma for a postdoc. What about you, Sam? Where are you from?
Well, I grew up in rural central Missouri. And so for those of you that maybe not know where Missouri is, it's right in the center of the U.S. I did my undergrad in Missouri. I got a master's in Kansas, right next door to Missouri. And then I did my PhD in Oklahoma. And now I'm a postdoc here in Oklahoma, although at different institutions. We don't talk about that very much.
What would you say is the biggest difference between Missouri and Oklahoma?
Oh my. I think for me it has to be the topography, the scenery, and the temperature. So it is much flatter here, and it is much hotter most of the year, I would say, in my experience.
I feel you. That is probably true from my Austrian experience as well. So Austria, where I lived, is not as flat and way cooler.
Yeah.
You said different institutions, right? So what is the institution you're in now?
Yes. So at Oklahoma State University, so that's where I currently am, we both belong to the Yushahed lab. So the PIs, the big bosses in this lab, Noha Yusuf and Mustafa El-Shahed, those are our lab PIs. And so they're big into the world of all things anaerobic microbiology, but primarily, and there's a heavy focus in the lab on these anaerobic gut fungi, which sort of leads me to you, Julia, and your research interests, because that's sort of your area of expertise.
That is true. That's what the postdoc is that I came for, is working with Dr. El-Shahed and Dr. Yusuf, who are very well known in my field of research. And I'm mainly focusing on anaerobic gut fungi, as Sam said. And for those of you who don't know, anaerobic gut fungi are the only phylum of fungi we know that is obligately, that is strictly anaerobic. So oxygen is completely toxic to those fungi. And it's not a well-known phylum. We don't know much about them, but we learn every year, and especially in the Yusuf El-Shahed lab, or the Yushahed lab, as you heard us call it, there's a heavy focus on those fungi. But not only, because we have Sam with us. So what are you doing, Sam?
Well, I always like to tell people that the lab is split into hemispheres, although it's sort of disproportionately split, in my opinion. I am more on the bacterial side of the fence, but essentially I'm I'm an anaerobic microbiologist by trade. Love growing things, particularly bacteria. I came up to OSU to be a part of what's called the MCCF. And yeah, I find myself up here at OSU these days.
Nice. And the MCCF, could you expand on that further maybe?
Yeah, certainly. So the MCCF, as we affectionately call it, is the Microbiomics and Culturomics core facility here at OSU. It was recently started about a little over a year ago, I think. And essentially what we are is we are a core facility that sort of, as the name implies, we focus on sequencing things and then on growing things on an as-need basis. And what we do primarily is we're responsible for helping out and stewarding 5 younger tenure-track assistant professors and helping them with their projects. Some of them there's a heavy need to sequence a high volume of samples, or perhaps they need to grow particular microbes from specific environments. And at the MCCF, we sort of help them to do those things. But in addition to just helping them, we also help out other people. We've got ongoing collaborations now with individuals that are not at Oklahoma State University. We've got collaborations with a few different institutions here in Oklahoma, and also now we're working on getting into places even outside the state of Oklahoma.
That's interesting. So anybody can contact you guys and say, help, I need some sequencing. I need you to culture this stupid bacteria.
Yeah. No, exactly. Yeah, we would be just the place for that. If there's a specific bacterium you want to attempt to isolate from a particular sample, we can help attempt to facilitate that here at the MCCF.
Nice.
And I guess so it's not just me at the MCCF. It's set up with Dr. El Shahed as the director. Then we have a core manager in Dr. Ayman Yassin. And then we have me. And then we also have a couple undergrads from time to time. So shout out Ty Young. If you're listening, go Pokes. Go Ty.
Go poke Ty.
Yeah, don't go poke Ty. Don't do that. I need him to run some gels later.
That's interesting. So I guess just out of interest, what kind of instruments do you guys have?
Yeah. So I will say, perhaps thankfully, that a lot of the instrumentation and the purchasing came prior to me arriving at OSU. But we have, I think, most of the standard lab equipments that you would expect from a core facility. But specifically, some of the stuff we have that would set us apart are the different technologies that we have to study microbes sort of across oxygen gradients, and those being like lots of chambers and glove boxes, whether they're a hypoxic chamber where we can control the concentration of oxygen to a particular percentage, or say we want to move into like an anaerobic chamber and be totally devoid of oxygen. We've got those capabilities now to do things with.
Saying chamber, for everybody who's not an anaerobic microbiologist, and even some that are, because I certainly came from a lab that did not have an anaerobic chamber, even though we were doing anaerobic work. What the hell is an anaerobic chamber?
Well, an anaerobic chamber, I tend to tell people that it's like a very large, sofa-sized vinyl balloon that has armholes. And inside this giant balloon, if you will, we fill it up with anoxic gases. And so these can be things like nitrogen, carbon dioxide, hydrogen. We'll vary the proportions of those kind of depending on the need of the microbes in question. But essentially, it's a giant, flexible, in our case, vessel that we can put samples, and we can put all of our consumables and things inside to keep them in an environment that's totally devoid of oxygen. And so we'll reach through, there's like special gloves and arms that we can then articulate and take and move things around and work again in an environment that's totally absent of oxygen. And so that's what I would describe our chamber as, is a big, expensive, thick balloon hooked up to some compressed gas tanks.
That's a pretty picturesque description. If any of you are interested to see actual pictures of our labs, we do have different social media accounts. The MCCF facility has an Instagram account, a Facebook account, a LinkedIn account.
Yep.
Did I miss
that
anyone?
we do. No, those are the three that we currently have right now. You can check us out on Instagram, Facebook, or LinkedIn.
And Ayman and Sam are both very active in giving you insights into the lab and our life in the lab.
Yeah, we do. We've been trying to post weekly. So you can expect about one solid picture from us a week with a little story. Usually it's microbiological. Yeah. So Julia, speaking of all things research, obviously you're interested in the AGF, the Neocalammasticomycota, as some might call it. I know, I got it right every once in a while. What are your general research interests? Is it all just AGF, or are there other things that exist out there?
So I'm very boring. I'm AGF all the way through. For the past 6 years, I've been focusing on AGF. I've been lucky enough to be able to do that. And the research field itself is so diverse, and I was dabbling in so many different aspects of research on anaerobic fungi, from the applied side to the basic research side, from trying to cultivate and preserve them to doing diversity studies in environmental samples, to even have an applied biogas reactor kind of thing going where we try to be able to grow them in biogas reactors. So this diversity has helped me to not get bored in the past 6 years. And I've been really enjoying my time, and I hope to continue for a couple more years. But what about you, Sam? What is your research focus?
Yeah, well, sort of in the vein of the MCCF, it's a lot of sequencing and it's a lot of culturing. I think, deviate a bit more than you in a sense that I'm not strictly anaerobic. But I deal with aerobes, anaerobes, the microaerophiles, the facultatives. But generally, it's a lot of growing things from specific places. Someone will want to grow bacteria from a wastewater treatment plant, or they'll want to grow a certain type of bacteria from a beetle, or maybe it's a soil sample. So I think in that regard, for me, there's a lot of diversity in the things that I do, but at the core, it's all about trying to bring these cultures to life in the lab.
Nice.
Yeah.
And you're certainly good at that. Also at collecting samples.
Well,
for those of you who don't know, Sam is our sample procurement specialist, and he earned that title.
And perhaps we can tease a future episode on annoying people and convincing them to give you weird things. But I do enjoy the sample collection process. Yeah, definitely.
It shows. We have freezers full of poop in the lab.
I did not handle all of these myself. Sometimes it's convincing people to give you tubes full of poop, not collecting the poop yourself. Although, again, sometimes both things are true.
Do you have any other hobbies than collecting poop?
Oh my, you mean like in the lab, or when I'm not in the lab?
I guess when you're not in the lab, since we will be talking a lot about in the lab stuff, let's talk about outside the lab stuff.
Yeah, definitely. So contrary to popular belief, postdocs do venture out into the sun every once in a while. We don't light on fire quite like vampires do, but it is an oddity to see a postdoc out in the wild.
We do get sunburned very easily.
Apply that SPF. But on a serious note, I would say my biggest hobbies are cooking and kayaking, and specifically I love to grill. I really enjoy grilling food, and kayaking out on the lakes, on the rivers. That's probably my most favorite way to spend my free time when I'm not in the lab. Yeah, what about you, Julia? What are your hobbies outside of the lab?
Well, I mostly have the same hobbies that I have inside the lab. I love exploring, so that's why I do research, and I love exploring outside the lab as well. I do a lot of hiking, I do a lot of driving around, especially here in the US, and I also do a lot of yoga.
Yeah, so we've got driving, we've got yoga, and we've got hiking. So have you traveled much inside the US?
I actually do, yeah, I did. At that point, I can say I have traveled a lot. I've been to California twice. I've been to Wisconsin. I've been all around Oklahoma, trying to get in as many hikes as I can. I've visited Kansas and Arkansas, and I will travel to Arizona soon as well. So yeah, I've been traveling a lot, not a lot as you probably have, and given that you've already lived in 3 different states.
Yeah. Have you been on the East Coast much?
Not in the last couple of years, but I think it was 10 years ago or so, I've been to New York City, but the East Coast is a blank slate for me. I still have to go there at some point.
Yeah.
Have you ever been to Europe, Sam?
I have not been to Europe. I've only ever been to Central and South America. I've not yet made it across the pond.
Interesting. What brought you to Central and South America?
Oh my! Well, I did a semester abroad in Ecuador, and so that was more or less my time in South America. The reason for that is, as I'm talking to you today as a microbiology postdoc, but there was a point in time in my life where I wanted to be a high school Spanish teacher, and so that's essentially how I wound up in Ecuador. And in Central America, I spent my honeymoon in Jamaica, and so I've sort of been Jamaica and then a little bit in South America.
Nice. What made you change your mind from high school Spanish teacher to digging around in poop?
Oh my, that's a whole nother episode in and of itself. Stay tuned for that, folks. But I think for me, it was like I've always enjoyed learning and I've always enjoyed talking, and I needed to figure out something that would allow me to make money and also for me to be good at whatever that thing was. And so I figured, like high school teacher, students are confined to their desks. They're forced to listen to me. I have to talk. It's great. It's a win-win for everybody, minus the students. And I was probably a sophomore in my undergrad, and I was in an education class, and I had sort of expressed interest in, instead of being like a high school teacher, I'm like, I want to be a professor at a university someday. I think I'd like to teach kids that are 2 or 3 years older than high schoolers. And that's sort of when the education professor pulled me aside and she said, Sam, you need a degree in content, like if that's the case. Like you don't need a biology and a Spanish education degree. You need a degree in like Spanish or a degree in biology. And so at that point, my life kind of flipped upside down and I realized why some professors are perhaps not the best teachers. There's an educational component that's lacking there. But yeah, and so for me, then I fell in love very quickly with microbiology my junior year and sort of transitioned away from Spanish into the hard sciences, if you will. And yeah, I've been kind of fumbling down this microbiological road ever since.
That is quite the life story to go from high school teacher to postdoc. Sounds very interesting. We might have to have another episode of you giving us your most profound learnings from that transition. Oh
my, we can maybe do that. But yeah, I think that would be good. A more in-depth conversation with the both of us about coming up and landing in the current spots where we are. All the twists and turns along the way.
Oh yeah.
Yeah.
Which actually brings me to the next thing we could discuss is like what is that podcast that we're doing even supposed to be? Where we want to go with this?
Yeah, no, that's a fantastic point. And I think for us, it really boiled down to there not being a whole lot of stuff out there in the, shall we say, the public zeitgeist about anaerobic microbiology. And as, the biased individuals that we are, anaerobic microbiologists, we figured that it would be, an interesting thing to do to try to make these anaerobes in all things, Life Unculturable, more accessible and bring just a dash of entertainment and pizzazz to the way in which we talk about them, just to make them not seem quite as scary as they're often sort of thought of being. I feel like there's sort of a negative connotation with a lot of the difficulties of anaerobic microbiology and some of the complexities, which are certainly there. But I think for us, we'd like to make it a bit more digestible for most folks.
Yes. And also along with that comes the talking about it, because I feel like, and I don't know if you had the same experience as I did, but I feel like in our field, the basis of the methods has been published in the 1960s. And from then on outwards, everybody was just doing their own thing without talking about the nitty-gritty parts and also standardizing those parts across fields and across labs and experiments. So I feel like this could also be a good part why we're doing this, to like get the word out, to get talking.
I couldn't agree more. I know of several times, even during my PhD, going and trying to ask someone that wasn't my PI for help with a particular anaerobic microbiology problem. And it was always, the answers were different depending on who I asked. And they were like closely guarded secrets that nobody wanted to divulge. And so on that note, I'm not saying that I'm going to tell you all exactly how I make my roll tubes, because that's proprietary information. But, I mean, I probably will. We'll spill some beans. And I think we also want to have the beans spilled from other people, because we're certainly not the end-all, be-all in all things anaerobic microbiology, but we hope to and aim to have some guest interviews and to chat with some people in the field, be they experienced folks that have come before us or perhaps contemporary counterparts that are rising up through the ranks as we are right now. And so with any luck, we'll be able to fumble through this guest list that we've been putting together and try to, have some people on for some really interesting conversations, whether it's about their current research or, perhaps a previous publication of theirs, or you mentioned there's plenty of literature from like the 1960s and 70s that detail techniques that we still use today, but the figures are hand-drawn and they're kind of hard to access. And so I think spotlighting some of that work, whether they're old publications or new publications, and kind of walking people through them and chatting about them in a semi-informal way, I think that would be something really fantastic to have.
Yes, I couldn't agree more. So it's kind of a platform for the community that we're trying to build and have like discussion about research and stuff, but also maybe because research alone is unfortunately not only research. There's so many strings attached. You have to do so many other things as a researcher. So I thought we could maybe also talk about that a bit and try to detangle some of those strings and maybe have a discussion about them as well, right?
Yes, I like the way that you said detangle those strings, because I'm imagining a very tangled set of wires right now. Yeah, whether it's balancing all of the stresses of going through your PhD, applying for jobs, writing manuscripts, getting manuscripts back in from review, finalizing stuff that your PI needs for a grant proposal, ordering that next box of pipette tips because the lab grinds to a halt without that. You're right. There are so many things that go into like the lab life, as it were. We could potentially spotlight a little bit of the lab life in addition to the hard sciencey stuff as well.
And on the same note as that, with the MCCF core, there are so many new strings that come into building a core facility. So that could also maybe be interesting for people outside, just to get a look at what it is like to build and work at a core facility and stuff like that.
Yeah. So I think making anaerobes approachable, sort of educating and entertaining, a little bit of science with a lot of personality. I think those are the types of things that I think we hope to accomplish here on the podcast. Yeah.
I agree. That is a very good summary.
Yeah. Yeah, and before we dive into something else, folks, Julia and I were planning on interjecting with a couple of little bits here and there. These may be recurring bits. We may scrap them after this first one falls flat on its face. But one of the bits that we thought might be interesting to do is something called Lab Yap, which we basically are going to be picking a topic at random. It may be sciencey, it may be not, and just kind of chatting about it for a few minutes. And so today, brought to you by Lab Yap, is Fahrenheit versus Celsius. I will say, and I will admit, as an American, that water freezing at 0 and boiling at 100, genius. Love it. The fact that the rest of the weather in my life falls between 25 and 35, I need more. I need more. It's always 30. There's no deviation. It's like so tight.
Well, you have to be fair here. You live in Oklahoma. So if you live at another place in the world, there's more deviation. But here it's like 30 to 45. That's it.
Yeah. Yeah.
But maybe because you started out so civilly in acknowledging the good thing about it, I can acknowledge that in Fahrenheit there is indeed more deviation in the everyday encounters you have. So you have like 50 and you need a jacket, and 90 and you shouldn't go outside. But yeah, I can't wrap my head around it. It's been 2 years or almost 2 years, and I still, like, if I see the weather report with like 78, I'm going, oh, okay, what am I to do with that? I guess I'm going to boil to death at 78 degrees Celsius.
I was going to say, at Celsius, that'd be a tough time. But 78 degrees, I'm thinking that's nice fall weather. Well, perhaps not deep into the fall, but that's nice weather.
In Oklahoma, maybe.
Yeah, in Oklahoma. I should clarify, folks. In Oklahoma. Yeah, no, I have the same exact experience, even with the opposite, with like the lab temperatures. It's like you learn the obvious 37 degrees Celsius because that's one of the important ones. And then from there, it's like, well, room temperature, sort of hovers around 22-ish. And then 45, 50 is the hot one. And then beyond that, I've got nothing. Absolutely nothing.
Well, yeah. I don't even know what to say to that anymore. It's like Fahrenheit does not make sense to me.
Yeah.
But it's fine. If you're happy, I'm happy.
Yeah, and I think that is a perfect way to end the lab yap is if you're happy, I'm happy, because I feel more or less the same about Celsius. Yeah. So I think hard science-wise, Julie, about the only thing that I haven't asked you yet that I wanted to ask you, just topically, again, we can, in later episodes, we can kind of go deep into the weeds and drown people in the water. But what are you working on right now? Like, what's going on in your lab life?
Oh, wow. Okay, we're going to tease a bit about our upcoming publications. So we have two publications coming up, hopefully at some point. We're currently, on the one hand, working on 3 novel genera of anaerobic fungi that we found in the tortoise guts. And we've isolated them and described them. And they kind of changed the taxonomy around a bit in the phylum Neocalammasticomycota. So that's one exciting publication. And then the other one is with the Anaerobic Fungi Network and some collaborators in Austria and Germany. We are trying to come up with a standardized protocol on how to deal with amplicon sequencing data pertaining to anaerobic fungi, because they are kind of difficult to detect and special in their own way. And we're kind of trying to come up with a standardized protocol and present that to the community that wants to research anaerobic fungi, so everybody can just have a go if they want.
Yeah. Now you mentioned the Anaerobic Fungal Network. Is that is that a large organization? Is this how many people are out there delving into the world of AGF?
So I would say it's double the size it was 3 years ago.
Whoa.
But it's like around 200 people, maybe, worldwide.
Wow.
So the Anaerobic Fungi Network is a collaboration of scientists. It's led by Joan Edwards, who is, if you are in that science bubble, you probably know her because she's a pretty iconic figure in the anaerobic fungi world. And yeah, it's just we come together. We have webinars that we have around quarterly, where also Sam joined us in one of those webinars. And we organize anaerobic gut fungi specific conferences and workshops and stuff like that, and just come together and try to foster collaboration between all of us, because there are only a handful of labs worldwide working specifically on anaerobic fungi, and even fewer labs that actually culture them. So in that kind of sense, it's not a big community, but it's a very collaborative and very fun community to be in.
Yeah, I like how you put that, and it was. I had a great time at my, one and only AFN webinar.
So far.
I was going to say so far. I'm going to have to get back. I'm going to have to get back into that. No, I really do appreciate you and Joan having me on, and it was fascinating to see. As you just said, there's only maybe 200 people, but it felt like so many people that were in that that call as it was live, and we were recording and chatting. But it was nice to see like people with that kind of common interest coming together, and from all parts of the world too. Like it wasn't just people here in the States; it was people in Europe and in and in Africa and in just all over the place in Asia. Like they're just labs and individuals. It might have been in the middle of the night for them when this talk was happening, and and they were still there and they were still excited and asking questions, and I thought that was really incredible. I really enjoyed that.
Nice. We loved having you on, and we will have you on again at some point.
Oh my. I will be back. I'll have my people call your people.
But that brings me maybe around to you. Like, what kind of research projects are you working on currently?
Yeah, that's always the $1 million question. And I usually tell people that I split my time about 50-50 between the MCCF, so the core facility, and then all the other 50% sort of doing other things that aren't necessarily core facility related, although I tend to lean into culturing bacteria in both aspects. So, for instance, in the core facility right now, we recently just wrapped a project where we were doing colony PCRs. And for those of you out there that maybe are like, what in the world's colony PCR? We're essentially just taking a bacterial colony and trying to give it a tentative identification. We had an individual that he had a strain library of about 1,500 bacterial isolates.
Jesus.
Thankfully, they were small Petri plates. It only took up 3/4 of a fridge. Not 2 entire fridges. And Iman and I went through, and we, in a high throughput manner, were able to give tentative identifications to those 1,500 isolates. And so that's like one of the things that we'll do in the MCCF. We recently just wrapped a project there too, where we had a person here at Oklahoma State that was very interested in the microbes associated with a particular type of beetle. And so he came to us and said, hey, you guys like to grow microbes. I also like microbes, but I don't want to grow all of them. So why don't you grow 500 beetle bacteria for me? And I had to like double check that it was 500 and not 50, not 5, carry the 0. And so essentially we set ourselves to task with a plethora of different conditions and media types and screening various colony morphologies, and eventually gave this individual 500 different, well, not necessarily different, but we gave this individual 500 strains for them to do, whatever they wanted to do with. And so it's a lot of high throughput, like growing and sequencing on the MCCF side. On the sort of the other side of that fence, just as, I guess, like a typical postdoc instead of like the MCCF scientist person, I'm very much interested in, again, anaerobic microbes, but I find myself in a season of research where I'm not super heavily invested in anaerobic microbes. So I've got a project in the works right now where I am attempting to grow some myxobacteria. And so for those of you that may not be aware of the myxobacteria, they're kind of an interesting, at least interesting to me, group of organisms that are kind of unusual. They're rather social. They're rather cannibalistic.
Those two things can't fit together.
Oh, but they so do. They so do. They're predominantly found in soil. And in one of the cool ways, I think, at least to me, that we grow them is we grow them using this bait plate method, where we essentially put a bunch of E. coli out on a plate where there's really nothing else, just water and agar and some salt. And we just say, go get it. And the myxobacteria will quite literally just swim out of the soil sample and start consuming the E. coli and just predating them. And so that's sort of one of the projects that I'm involved in right now. And then the other that would probably be more anaerobically inclined is working on describing some novel bacteria. So novel, not in the sense that it's like brand new, never been seen before, but it's just sort of not yet been cultured. It's always existed, but we just haven't been able to grow it.
Unculturable.
Yeah, it was Life Unculturable until Sam Miller came along. and started looking at blue whale feces. But yeah, so we got in from a collaborator up in Wisconsin. Shout out Richard if you're listening. Some blue whale feces, and we started an undergrad and I started to do just like a sort of a culture dependent analysis of what was going on in there inside the chamber, and we sort of hit pay dirt in a sense that we found some what we perceive to be as like kind of cool anaerobic microbes, and so we're in the process of working through that data now. And so that's sort of what I'm into on a daily basis in the lab.
Wow.
Yeah.
Blue whale feces.
Yes. Yeah. If it poops, it scoops. We're interested in it all, folks. If you've got a tortoise in your backyard, we want it.
Oh yeah.
Yeah.
If you have an iguana, we want it too.
Exactly. Yeah. If it's
or some rodents.
Yeah. Exactly. If I guess I should clarify. If you're listening to this and you're thinking we should send Sam and Julia some of our, and by our I mean animal feces.
Herbivorous animal.
Yeah, herbivorous preferably from a smell perspective. If you don't sing this animal in the first couple of verses of like Old MacDonald Had a Farm, like we're probably good on sheep and cows and that kind of stuff. But yeah, if you've got an iguana, if you've got a tortoise, a platypus, maybe you live in Texas, you've got some kind of exotic animal that no one else in the world has, we'd love to have it. We are always
poop, though,
always
not
interesting.
the
Yes,
animal.
yeah. Please don't FedEx us like a chinchilla. We would we would prefer that it just be chinchilla feces. So just to clarify, folks, I don't think FedEx is going to be happy with that. And we can probably talk about this more when we do the sample collecting episode. But I had an individual recently offer to just send me like whole fish.
What?
Like they were going to ship me live fish.
I mean.
Yes.
Didn't you say you like to grill? So we could just could have a lap party.
Gee, I do, but I'm like we gotta work something else out. So I don't need a pallet of fish arriving at the lab door. Like, what have you bought this week, Sam? Well, technically it was free. I paid for shipping.
We're on the death note list of this FedEx guy.
Oh yeah, yeah. It doesn't matter what shipping agency you are. We're blacklisted. So speaking of blacklisted, this is a horrible transition, but I'm going to make it anyways. We haven't talked about the culture collection that you manage in the lab. So you kind of mentioned earlier that you're in the process of describing a couple new genera of AGF from tortoises, but I think for me, when I got into the lab, one of the coolest things that I quickly realized was the magnitude of the culture collection that we possess in the Yusha Head Lab, and the fact that you're more or less like the sole person responsible for making sure that it doesn't just keel over and die. And so what's that like? So I guess for the listeners out there, like how many AGF have ever been grown, and how many do we have that you take care of?
So in total, we know in this phylum of Neocalamystigomycota, we have 22 genera. And I think something like 40 species. I'm not sure on the species count, but that was the total of genera grown so far. With our 3 new ones, it's of course up to 25 genera. And 13 of those 22 have actually been first isolated, first described in the Yusha Head Lab. Of the 22 genera, we currently have around 13 genera growing. Not the 13 that we described or that the lab described, because some of them really died on us. They like to do that. But 13 genera out of 22 so far. And those fungi, they are really fastidious. You have to transfer them twice a week in fresh media, otherwise they die on you. And we keep them in duplicates, so we always kind of have a fallback in the second duplicate. And yeah, so that means around, I think, 55 tubes I'm transferring every week. And I do have to say I'm very fortunate to have a lot of undergrads that help me with like media production and transferring, not only undergrads, sorry, Sakalia, also PhD students who help me. I'm so sorry. So I'm very fortunate not to have to do that alone because it would be a lot of work alone.
Yeah, because that's so it's 55 tubes twice a week.
Twice a week.
Yeah. At 10 mils a tube. That's a lot of media to make.
Yes.
Every single week, and it never stops. No holiday for the culture collection.
It never stops. Yeah, we have to coordinate holidays and stuff like that because it doesn't stop. And they do not freeze well. I would know. I did my PhD on it. So there's no reliable preservation method so far.
So far. Wow. And so all of these isolates that you have in the culture collection that you're taking care of, have any of them come from other labs? Like, have other labs sent you strains? And
no. So these were all from our lab, as far as I'm aware. I've inherited some of them, so I'm not 100% certain, but I'm 99% sure they were all isolated in our lab, because also they do not freeze well, and they also do not transport well. They do not like being cold in any kind of way, and they are very much Oklahomans in the sense that they like 37 or 39 degrees and nothing below that.
Wow. Okay. No, that's so interesting. I know for me, from the bacteria guy perspective, I would have something on a plate, or it would be in a tube, and I'd probably just toss it in the fridge, leave it for a month or two, and come back and just hope it works.
I hate you so much. I hate you so much.
Because I'm thinking now, like one of the projects we were working on in the MCCF, it was this guy that was interested in mosquitoes, and specifically mosquito midguts, and we were growing bacteria from the midguts of mosquitoes, and because why not? And I had some isolates in the fridge, and I think they've been in there since probably June.
No.
So June to last month was September, and I just went in a few weeks ago because he asked me, like, oh, do you still have, isolate 12 and 11 and whatever the numbers were? And I went, yeah, I'll go check. If I have it, I'll see if it still grows, and it still grows. Yeah.
You're just hammering it in.
I know. I'm trying to tell you. Prokaryotes greater than eukaryotes, folks. Come on now. And that's just crazy to me too, that there's not I don't want to say a better way, not like what you're doing here isn't better, but you can't just contact like a culture collection and say, hey, we want to add this to your stash of AGF that you already have.
Yeah,
because they just overall don't preserve well. That's so fascinating. Yeah. Wow. Yeah.
But on the other hand, I'm glad that it works so well for you with like keeping them in the fridge, because imagine having to take care of 1,500 isolates.
Yes. that would have been rough.
It's all fair and square here.
Yeah. Wow. And you mentioned earlier when you were talking about the AFN that you're working on ways to like standardize sort of informatics to look for these. And I get, because I've listened to you talk about this a couple of times now.
For you.
No. And that's probably a conversation worthy of its own, 30-minute segment. But just generally speaking, so like I know for me, if I'm looking at, diversity surveys of bacteria or something like that, like I'm looking at the 16S rRNA gene. That's my, gold nugget. Like, do you do something similar with the AGF? Do they have like a gene that you target?
Yes, we do. Unfortunately, it's not like with fungi in general, like not just AGF, but general fungi. The pendant to the 16S would be the 18S, but that unfortunately does not work very well for fungi in general because it's too conserved in most of them. And so the fungal world has moved to something called the internal transcribed spacer region, ITS. So if you have the rRNA operon, it's like first it's this 18S or the 16S in bacteria, then comes the ATS1, then the 5.8S rRNA, and then the ITS2, and then the LSU. So the fungal world in general is all riding the ITS1 train. Some are riding the ITS2 train. But what we've moved to, or we're forced to move to, is the LSU, so the large ribosomal subunit. And the start of that ribosomal subunit gives us the best taxonomic resolution in diversity studies.
And so it would be pretty similar then, in that you would sort of amplify like a portion of that, and then sort of compare it to other things to get a feel for your community structure.
And that is also what the AFN has been providing for the community in general, is a database, a reference database that you can use. Like if you amplify your LSU, or we even have the ITS1, but we don't recommend using it, then you can have that reference database that the AFN provides to check out what fungi you have in there for your analysis. Similar to what Unite is doing for the general fungal world, and Green Genes. And what is the other one? Green Genes and...
let's see, Green Genes.
I know that...
Big database.
RDP? Silva. That was the one.
I knew I had a database in the back of my brain somewhere.
I kind of threw that question at you, but yeah. So the AFN provides an anaerobic fungi-specific database as well.
Okay, that's cool. And another little segment that Julia and I were potentially flirting with the idea of doing is like a questions from the audience. And so, as we obviously have the #1 anaerobic microbiology podcast on planet Earth right now. Thank you, Mom, for listening. We don't have a whole lot of questions from listeners at this point. So we did what any good postdocs would do, and we asked the undergrads in the lab to, if there were any questions that they might want answered here on this internationally renowned podcast. And of the fistful that they came up with, I think the one that was the funniest to me, the best, was if you had to summarize your research activities with like a song, what would that song be and why? And so do you want to take this one first, or do you want me to take this one first?
Sure, I can go first. That was such a good question, and I actually thought about it a lot overnight, and there's so many things that come to mind. Like if you're a PhD student, your song would probably be Highway to Hell, hands down. In anaerobic microbiology, it might be Under Pressure by Queen, given that our tubes are seldomly not pressurized. But I would say, for me, my soundtrack probably would be a more recent one, and it would be Vacation by the Dirty Heads. But I don't even know if you know that song, Sam, because your music taste is something else.
Well, it's just earlier than the present is what my musical tastes are. And I like that you put the Highway to Hell reference in for the PhD students. I was just thinking Bon Jovi, like Living on a Prayer. You're like, oh, I'm halfway there. I'm not going to sing it, folks. I'm not going to sing it. It's not that kind of podcast. But yeah, I think there's so many different lyrics that you could probably take out of context that would fit a portion of what we do or how we do it. But I think for me, if I had to pick just one song, as you've already alluded to in my musical choices, because I'm fascinated with sort of 2000s pop. And for me, I think the choice would have to be Avril Lavigne's Complicated. Again, I'm not going to sing it, but it's, why'd you have to go and make things so complicated? And so, again, making anaerobes approachable, there is complexity involved, much like there is with anything. But I think, for me, when I think about that song and I think about what we do in the lab, oftentimes there's a lot of babying around that comes with like the anaerobes. Like there's a lot of extra additives and goodies that have to go into making the media. Sometimes it's as simple as putting, so many grams of TSB powder in a liter of water and autoclaving it and you're good to go. Other times it's not. It's handling all of these 14 different chemicals to make a salt solution that you're then going to filter sterilize in after it's autoclaved, after you've made an anoxic vitamin, solution and an anoxic antibiotic stock solution. And you've got all of these different things that have to go in at particular concentrations and proportions and adding rumen fluid and sometimes blood. And sometimes it just gets complicated. And so for me, I think that's the song that holds true to myself right now in this moment. Maybe one of these days I'll get more into whatever that song was you just mentioned.
No, I feel you. I feel you. I can see where you're coming from.
Yeah.
And I'm gonna play the song after we turn off the podcast for you, so you also know.
Yeah. I mean, eventually I might branch out and, listen to something that's post 2013-ish, but if you find me in the lab, I've got my headphones in, or I'm the guy that's got the speaker going, and yeah, it's going to be pop divas from the 2000s. So, yeah.
I love the energy in the lab because you bring that energy, that pop diva 2000 energy.
Yeah, and I will say that's another thing, kind of going back to our PIs, Dr. Yusuf and Dr. Elshahed, that positive energy. And I think at least I feel fortunate in a way that, the other individuals in the lab, like, they're fun to work with.
Yes.
I think we've got a really good group of students, both undergrads, graduate students, our, core facility manager, Iman. There's a really good group of not just scientists, but human beings.
Yes.
And it's fun to every once in a while kind of let loose and, have a picnic out by the lake, or not the lake, the pond. It's hard to call it a lake. It's a pond. And, get some pizzas in, share a couple of laughs before we go in and troubleshoot why a PCR failed. So I think that's kind of a really nice point about being in this lab. I really do enjoy I hesitate to call it the culture, but it's, this one's culturable. Yeah.
I totally agree. And I also that it's like all of us are very different from one another, but that kind of makes it pretty cool because you have like different perspectives and different ways to interact. And yeah, it's just overall a good vibe.
Yeah, no, I couldn't agree more. And I really do enjoy that. I think, in a lot of ways we get to bring out the best in each other, as corny as that sounds.
Aww.
I think we get to do that.
Yes.
In the lab.
Absolutely.
Yeah.
I love that. I love that sentiment. We could have it as the outro right there. Like, podcast done. See you next week.
See you next week. Yeah. Like and subscribe, people. Like and subscribe. And let me think. I think the only thing that we didn't cover that I was hoping we would cover is just a why anaerobes. We've kind of delved into how we got into anaerobes and then even some of the research that we're doing and some of the people around us, what they're doing. But what specifically, if you had to give people something to like take away about anaerobes, whether it's, a fun factoid or, a tidbit or a bit of advice, what do you think you would leave them with?
That's a good question. I guess I have two kind of things, but the most important one I would want people to know is that it is not as complicated or as difficult as you would feel it is. At least for me, that was true. And I don't know if that was true for you as well. But before I started and just reading about it, was like, how are we going to exclude oxygen that is all around us? And I couldn't imagine how that would work reliably, but it is working very, very reliably, I have to say. And it's not as difficult as I made it out to be, although it is. You have to have equipment to do it most of the time, but also not as much as you would think. You can even do it without an anaerobic chamber at some point, like we did in Austria. But yeah, so that would be my most important takeaway. And I forget what the second one was, so I'm just going to give it back to you, and you're going to give us one of your takeaways.
Yeah, fair enough. No, I like what you said, even though I, ode to Avril Lavigne's Complicated being my theme music for my research activities. I do think that some people perceive anaerobes to be almost impossible to work with just because, like, oxygen is everywhere, and like as soon as the chinchilla poops, like there's oxygen and there's oxygen in the tube, and like how do you get anaerobes from point A to point B? And so it does come with its own set of limitations and difficulties. But I would agree also that it's not that bad, but you do need this special equipment, that big oftentimes, it really helps to have like a chamber or a gassing station or, with a manifold and to be able to switch around gases and do things like that. And so I think oftentimes, like you said, it's a big equipment crisis more so than it is like a techniques type crisis. Like if you're a good quadrant streaker outside the chamber, you're a good quadrant streaker inside the chamber. But I think for me, if I was going to come up with my own on the spot here, it would be that, as biased as I am, I would say that anaerobes are important. Whether we're talking about anthropogenic things that we really enjoy, whether it's cheese, beer, or bioethanol. Yeah, kombucha, like all the things that, well, not all the things, but several of the things that we just sort of enjoy and take for granted. Those anaerobes play a part in those processes, whether it's making them or after you've consumed, say, some yummy cheese or some kombucha, in digesting them. I think anaerobic microbiology sort of, for me, it pops up in more places than people might think. And so I think if you look hard enough, you find an anaerobe somewhere. Yeah.
That is spot on. Another outro right there. If you look hard enough, you find an anaerobe somewhere.
Yeah.
Well, nice. Yeah.
Well, this was an absolute blast, and we look forward to chatting in your ears very soon.
Yeah. Thank you for listening. Keep an eye out at the MCCF platforms for the next podcast episode.
Yeah. As we work on putting together a social media presence.
Let's call it media presence.
Media.
Let's be real. Social is not our thing.
Exactly. So I suppose this is where I'll sign off and say, bye, everybody.
Bye.